I'm going to discuss a region of genetic code in E. coli called the Lac Operon, which is used to metabolize lactose. The key regions of the Lac Operon are the repressor, LAC-I, the gene encoding for the Lac repressor protein, the promoter, LAC-P, which is the binding site for RNA polymerase, and the operator, LAC-O, which is the binding site for the repressor protein. E. coli break lactose down using three enzymes: beta-galactosidase, which is encoded by LAC-Z, permease, which is encoded by LAC-Y, and transulcetolase, which is encoded by LAC-A. Beta-galactosidase converts lactose into glucose and galactose. Permease transports lactose into the cell, and the function of transulcetolase is not currently known. One last thing to note is that polycistronic RNA is produced during transcription, and that while P and O are regulatory regions and only affect the genes that they are sitting next to, they act in cis. I codes for a diffusible regulatory protein, and it can affect both chromosomes, or it can act in trans in a partial diploid. Let's go over the mechanism in a wild-type Lac Operon in the absence of lactose. Our genotype is I+, P+, O+, Z+, Y+, so all of our genes are functioning. We're specifically going to think about the concept of negative inducible control. Without lactose, the repressor protein is going to bind to the operator and block RNA polymerase. Without RNA polymerase, we cannot produce beta-galactosidase or permease. So you can think of this repressor protein as the brake on a car. There's no movement even if you press the gas pedal because we've locked the tires. Now let's look at what happens when we do have lactose. So what's going to happen is lactose gets converted into allolactose, which binds to the repressor protein, making it inactive, and it can no longer bind to the operator. Now, RNA polymerase can now transcribe the structural genes, so we get production of beta-galactosidase and permease. Our brake has been shut down, and now the car can move. Let's look at an example of a partially diploid mutant Lac Operon. Here, for our first copy, everything is normal, except we have Z negative, so that protein will be defective. And in our second copy, we have a constitutive operator, which means it will never bind to the suppressor protein. It's always on, so our enzymes will be produced regardless. However, we have a dysfunctional Y gene, so we can't make permease. So let's see what happens when lactose is absent. When lactose is absent, we're not really going to see the effect of the faulty LAC-Z gene because we never get to that step. Our repressor protein works like normal and blocks RNA polymerase. Now with our other gene set, we have our constitutive operator, which never binds to the suppressor, so we can produce our enzymes. However, here we have a faulty LAC-Y, so we do not produce permease. So overall, in the absence of lactose, we can make beta-galactosidase, but we cannot make permease. Let's look at what happens when lactose is present now. So in our first copy, the allolactose is going to inactivate the repressor protein, and RNA polymerase is going to transcribe our enzymes. But we do have the defective LAC-Z gene, so we do not make beta-galactoside, but we do make permease. In our other copy, we have our constitutive operator, so it doesn't matter whether we have lactose, RNA polymerase is going to transcribe our enzymes. But in this copy, we don't have a working LAC-Y gene, so we can't make permease, but we can make beta-galactosidase. So overall, when lactose is present, with our mutant partial diploid, we can produce both permease and beta-galactosidase.